My name is David Deamer. I've been involved with nanopore sequencing for the last 30 years, and now I'm using it in my research on the origin of life. And what you're going to hear is that we can generate nucleic acids just by wet-dry cycling. I'm going to begin by giving about 10 seconds for you to read what Charles Darwin wrote to Joseph Hooker in 1871. Well, I think that Darwin was on the right track. He's talking about a warm little pond instead of the ocean, and he knows about proteins, but he doesn't know that they're polymers of amino acids. And nucleic acids hadn't been discovered yet, so he didn't know anything about that. Now, he was probably thinking about freshwater ponds in the English countryside, but the problem is that these lack thermodynamic properties that are necessary to have some of the reactions required for life to begin. And the most important of these is polymerization. So there is an alternative, and that alternative is an evaporating pond, a hot spring pond, someplace in Kamchatka, Russia. I was there for 2 trips in 2002 and 2004, and I took this picture on Mount Butnosky on one of those trips. It shows a puddle just sitting there after a rain, and you can imagine that that puddle is going to evaporate pretty soon. And that's what's missing from Darwin's idea. Unless you can concentrate organic compounds in the puddle, it's not going to work. We're not going to get concentration. So just imagine that this little puddle is evaporating, and all the stuff in it, all the organic solutes, are going to become very concentrated. So, thinking now back to 4 billion years ago, we're looking at the prebiotic Earth. And there's an atmosphere of nitrogen, carbon dioxide, but no oxygen because there's no plant life, no photosynthesis, no animals, no bacteria even. It's a sterile environment just waiting for something to happen. I might say that Mars also had shallow seas and volcanoes. Maybe life began on both planets. We call this a prebiotic analog. Prebiotic means before life began. And this is in California, in fact. It's on a volcano named Mount Lassen. And I've done quite a bit of experimentation at this hydrothermal field. And I'm going to claim that I think that this is a better place than the ocean for life to begin. And as I continue my talk, you're going to learn why. So we've been visiting other volcanic areas, Hawaii, Iceland, Kamchatka, and New Zealand. And these are all plausible models of the primitive Earth environment. And in particular, Kamchatka has recent volcanism that has sterilized it. So if I go to Kamchatka, I'm going to have a sterile environment to do experiments. Here's how I go to Kamchatka, and you can too if you want to. You hop on a plane in London or in San Francisco, and you end up in Anchorage, Alaska. And the next morning you hop on a Russian flight to Petropavlovsk. Petropavlovsk is on the peninsula of Kamchatka. It's the most volcanic region on the Earth. Vitus Bering was first to explore the Kamchatka coast in 1740, and he discovered a bay near the southern tip. He named the bay after his 2 sailing ships, St. Peter and St. Paul. In Russian, that translates to Petropavlovsk. So if you look at that image, it's an artist's rendition of a satellite view of Petropavlovsk. You can see Mount Butnosky just below it in yellow type. That's where we're going to end up. But first we got to get to Petropavlovsk. This is what it looks like as you come in for a landing. Mount Koryatsky is in the background, and you can just imagine how volcanically active this peninsula is. Petropavlovsk looks new, and it is new. Most of that built up in the last 50 years, because before that it was just a little village that happened to be named Petropavlovsk by Vitus Bering. So there is now an active tourist industry with helicopter service. So on our first trip, we flew to several nearby volcanoes, and we decided that Mount Mutnowski was likely to have hydrothermal springs that we could use to test Darwin's idea. This is what Mount Mutnowski looks like from up in the air, satellite view again, and we would camp about 2,000 feet below the summit on both of our trips, and we would hike uphill along a small glacier and look down into the crater. So we wanted to find a hot little puddle, and the first place we looked was in the crater itself. So here I am sampling a fumarole. Now this is kind of dangerous. Even though I had a 10-foot aluminum rod with a flask to get some hot boiling mud, all those fumes had more than water vapor in them. They had hydrochloric acid, little hydrofluoric acid. They had SO2, which is this sulfur compound that you can smell in all volcanic areas like this. Well, I took that mud back to home, analyzed it, and there were no organic compounds. It was sterile. Fumaroles like this were too hot for organic compounds to survive. So on my second trip, I decided to be safe. And I took my own prebiotic soup to add to a boiling 10-liter puddle on the flank of Mount Mutnowski, away from the crater. That white material I'm pouring in was a mixture of 4 amino acids, 4 nucleobases, myristic acid, a 4-carbon fatty acid. And that, by the way, is why it's white. It's a molecule that just sort of makes little particles that have that white, milky color. And then there's some glycerol and phosphate. The water itself was acidic, pH 3, because of sulfur dioxide that is an acidic gas. Well, I took samples minutes later and hours later and days later. I took those samples home to analyze. We actually published the analysis a couple of years later in the Philosophical Transactions of the Royal Society. So if you're interested in seeing the details, you go to our publication. But the important thing I noticed is a white froth appearing all around the edge. And I recall that the myristic acid is in fact a soap. It's a hydrocarbon chain with a carboxyl group at the end, and that makes it a soap. And in fact, it's one of the molecules that we use in our metabolism. So that white stuff is actually membranes that have been formed by this soapy molecule. And I realized that this pool was evaporating. And you can tell that if you look at the lower right-hand corner, there's a big white rock, but around the bottom it's kind of a reddish color where it's moist. And you see the other rocks there with the white on top and the moist. So this is evaporating. And it seems to me that that evaporation might add enough chemical energy to allow polymerization to occur. So what do we have to polymerize? Well, the one that I'm going to work with is nucleic acids. We're going to add nucleotides, the monomers of nucleic acids, and we're going to see whether we can get a real nucleic acid just by putting it through a wet-dry cycle. We also know from other people's works that proteins can be formed by wet-dry cycles. And from all of my own early research, we know that amphiphilic compounds like myristic acid can form membrane compartments. So that is what I have to work with. Now I need a little chemistry lesson. This is the chemical bond that we have to make if we're going to synthesize RNA or DNA from its monomers. The example I'm showing you here is ethanol added to acetic acid, and there's a spontaneous reaction called condensation, where an ester bond is formed and water is a byproduct. The compound that is formed is called ethyl acetate, and that's the solvent that is in nail polish and nail polish remover. Amazingly, DNA and RNA are classified as polyesters. They are linked together by ester bonds because there's a phosphate group there in the adenosine monophosphate to the lower right, and if you put it in solution with other adenosine monophosphates, there is a spontaneous, slow reaction that makes what we call the dinucleotide. And again, the product is water. Now, if we evaporate the water, we're going to pull that reaction to the right, and we're going to make more and more ester bonds. That, at least, is the principle that we're testing. Does it work? Well, I got home from my trips, and I made a homemade hot spring. This is a plastic container that's filled with carbon dioxide. It's got CO2 flowing into it through the little tubes on the right and left-hand side of that disc. The disc has wells where we put a glass vial with the reagents we want to work with. It's heated by those hot plates underneath to about 80 degrees C, and it rotates about once an hour by a stepper motor. There's also water feeding in. That little perfusion pump in the lower left has leads in, and as it turns, it gets rained on. Each vial gets rained on about once an hour. So to test the hypothesis, we're going to put a mononucleotide solution, 10 millimolar, adjusted to pH 3, which is the optimal pH, put it through multiple wet-dry cycles, we're going to get products. And then we're going to analyze those by mass spectrometry and nanopore sequencing. Well, here is what you get from a mass spectrometry. This is a collaboration with Richard Sayre in the chemistry department at Stanford University and his postdoc. The postdoc is named Xiaowei Song, and he's first author on the paper that we published just last year. So he did the interpretation of these peaks that you see. And as soon as he saw those peaks, he said, aha, I'm looking at a oligomer of UMP. So he knew that it had to be UMP, and it had to be an oligomer because it had multiple peaks in it separated by the mass of one proton. Each of those peaks is the same molecule, but with more or fewer protons attached to it. So he said to himself, well, I'm going to simulate a mass of 3,323 mass units, and I'm going to see whether I can find a simulation that matches that peak that you see there. And if you look to the right, to the left, you'll see a red square, and it turns out that if he imagines that this is a oligomer of 43 UMPs that has lost 42 waters, because you pull water out to make the ester bond, then you have to add a sodium and then 4 more sodiums to give it 4 positive charges. So that is an exact match, and he concluded that this was a 43mer of uridine monophosphate that we made by wet-dry cycling. And he didn't stop with just that one. He did lots of these. We supplied him with UMP, AMP, and deoxy-AMP and thymidine monophosphate. That's ribonucleotides on top, deoxyribonucleotides on the bottom, and they all turned into oligomers that he could separate. Well, we were very happy with that. Nobody ever tried something like before, so we wrote up a manuscript and asked a couple of knowledgeable colleagues to take a look at it, see whether they agreed. One of them said, well, that's very nice, but you had better do a separate analysis to prove to me that that is really oligomers of UMP and the other mononucleotides we tested. Well, that's where nanopore sequencing comes in. Karen Miga had just set up a nanopore sequencing facility at my campus, so I took some samples over and gave it to Josh Gardner, who runs the Prometheon there, and we tried TMP by itself, we tried DAMP mixed with TMP, and we tried all 4 nucleotides mixed together. They all made oligomers. They could be sequenced by the Promethean just as though they were biological DNA. I was really surprised by that because we just made these from scratch, from monomers, by putting them through wet-dry cycles. I didn't really expect it to work this well. We got hundreds of thousands of reads per sample. So we concluded that in fact mass spectrometry and nanopore sequencing results confirm that wet-dry cycles simulating hot springs associated with volcanic activity can drive polymerization. Well, what about Mars? Mars was very much like the Earth 4 billion years ago. We can still see water left over on Mars. That's a little crater on the left. It's got some frozen water because it had never been exposed to direct sunlight. Over on the right, you see layers of icy sediment that are on the edge of another crater. There was water on Mars 4 billion years ago. Life may also have begun on Mars. I want to send a solid-state nanopore device to Mars and have it look at melted ice and see what we can see. We might just detect preserved fragments of nucleic acids. So to sum up, the alternative to an ocean origin is that life began in freshwater hydrothermal fields, subjected to wet-dry cycles. That means that life did not invent nucleic acids. Instead, chemical energy from the wet-dry cycling generated RNA-like polymers or DNA. If amphiphiles are also present, the polymers can be encapsulated in vast numbers of membranous compartments that we call protocells. Each one is different from all the rest. It's like a microscopic experiment. Protocell populations can undergo selection and evolution. So the next step is to show that protocells that we are now generating have functional RNA in it, and we have some ideas on how to do that. Thank you.